wonder, if you have 100 random phage that infect gram-negative bacteria, how many of them are going to be homo-immune?
I've no idea. So the question is, if you have 100 different bacteriophages, all of which are capable of infecting the same host, how many are homo-immune? And for E. coli phages, although I don't remember the numbers offhand, it's relatively easy to isolate E. coli phages and find subsets that are homo-immune. If you look amongst mycobacteriophages, it's not a particularly high number. I think that out of about 50 genomes, 50 phages that have been well characterized, there's maybe 3 that share immunity and the rest don't. So it depends on how diverse that population of 100 genomes are, and that depends on what host you're looking at.
Well, the 3 shared, so the 3 that have a shared immunity, are they otherwise closely related?
So the question is, if you take 3 genomes that share immunity, are they otherwise closely related? And in that specific case, they are reasonably closely related, relatively speaking. It turns out that, just to make life a bit more complicated perhaps, that if you were to just look at the plaques of those 3 bacteriophages, 2 of them would indeed form turbid plaques that one would just look at those and say, there's a fairly standard temperate phage forms turbid plaques like we would anticipate. The 3rd forms beautifully clear plaques. It kills those cells more efficiently than almost any other bacteriophage that's been examined for that host. But they do show immunity in the sense that that clear plaque fails to form plaques on lysogens of the other 2. And the best way of thinking about it is that it is and genomically, we can see that it is a derivative in which a deletion has removed the repressor gene. So it can no longer make repressor, but it is subject to the action of repressors made by prophages of the other phages. One more, maybe? 2 more.
Your point that the population turns over every few days implies that something is removing phages from the environment, and that phages have a lifetime, a sort of a half-life in the environment. What are those factors, and what is the sort of a half-life of a phage in the environment?
I don't know I don't have a specific answer, but UV especially in the ocean samples where many of those experiments have been done, UV definitely plays a role. And there are some interesting implications that I don't have time to go into, but if it's been shown, for example, that some phage genomes, if you experimentally show them so that you have duplications of large segments of the genome, you actually get much better survival to UV because you have duplicate copies of the genome. So UV at least is one important factor. There's protozoans that specifically eat bacteriophages in the ocean as well, right? So there's probably several other factors that are helping to turn over the phage population. One more?
So for lysogens whose prophages are propagated extrachromosomally, like an episome, they must have their own origin and replication. And are some of those actually high copy? Are they multiple copies per cell?
All the ones that I'm aware of, P1 and N15, they tend to be low copy. Okay. They do have origins of replication, and most of them have, I think, fairly sophisticated partitioning functions to make sure that that the prophage is not lost, the extrachromosomal prophage is not lost at any significant frequency during cell division. So yes, they have origins of replication and they have partition functions.
Thanks.
I actually, we've observed some that have the bullseye morphology, so they're not exactly the clear-cut turbid or clear plaques. Can you speak to what's going on there at all?
So when one looks at bacteriophage plaques, there are those that are of a clear morphology, plaque morphology, meaning that if you look at the plaque, all the cells are dead. There are those that you can look at, and I showed you a couple of examples, where the plaques are unmistakably turbid. And those, I think in virtually all cases with all different types of hosts, are temperate phages, and stable lysogens can be readily recovered from those turbid plaques. Then there's a third group of phages, which are not quite so conveniently explained. And they form various different types of plaque morphologies. Some of them are sort of hazy, rather than being clear or obviously turbid, meaning there's some live cells there, what appears to be some live cells there, but looks distinctly different from true turbid plaques. And yes, you're right. You see phage plaques that have sort of halos around them, and you see those that appear to have rings and other structures inside of them. Almost certainly an abundance of really interesting biology, not much of which is that well understood. But I think that one way of thinking about these intermediate types of phenotypes is that we have to be careful about interpreting or extrapolating what we see on a Petri dish in the lab to what's actually happening out there in the ecology where these things actually exist. For example, when you see those different types of plaque morphologies, it may be that the host that you're growing on is a host for that bacteriophage, but it may not be the bacterial host which the phage has been most recently infecting in the environment from which the phage was isolated from. So maybe the bacteriophage that forms a hazy plaque in one bacterial host might actually be a very good temperate, turbid plaque former if you were to pick a different host. And so you have to be very wary about assuming what the host actually is when you think about it in terms of interpreting the plaque morphology that you see. Does that make sense? Okay. All right. We will continue then.